When you think about the immune system fighting off a disease, it is really tempting to picture this straightforward cinematic battle.
Oh, absolutely. The human mind just prefers that kind of, simple, efficient narrative.
Right. The pathogen shows up, your white blood cells recognize the invader, they attack, and bam, the threat is neutralized. It feels like this open field skirmish where the good guys just have to outnumber the bad guys.
And I mean, to be fair, in certain scenarios like fighting off a common cold, for example, it actually functions somewhat close to that.
But the moment you step into the world of solid tumors, that simple narrative just completely falls apart. You are no longer looking at an open field battle.
No, not at all. You're looking at a grueling, really complex, heavily entrenched siege.
Exactly. And today, we are doing a deep dive into a 2026 paper from Cell Reports Medicine by Rodriguez and colleagues that tackles, honestly, one of the most frustrating puzzles in modern oncology.
Right, which is how to break that siege using CRT cell therapy.
And for you listening, today's mission is to understand a genuinely groundbreaking strategy to fix this failure. And the solution involves hitting the tumor with a highly targeted radioactive smart bomb before the immune system ever even arrives.
Yeah, and the frustration we're talking about really stems from how successful CAR T cells have been elsewhere. For anyone following cancer research, CAR T cells are the undisputed heavyweights of blood cancers.
Because doctors can take a patient's own immune cells, genetically engineer them to become these highly specific assassins, and put them back into the body. Right?
Exactly. They have functionally cured certain leukemias and lymphomas.
That's because blood cancers exist in a liquid environment. The engineered T cells can just, circulate in the blood, find the floating cancer cells, and eliminate them.
Right, they just bump into them. But solid tumors like neuroblastoma, which is this devastating pediatric cancer that the paper focuses on, they are entirely different beasts.
When these elite CAR T cells face a solid mass, historically they just fail.
They do. And to understand why that smart bomb you mentioned is necessary, we really have to look at the anatomy of that failure. The researchers zeroed in on two primary reasons. CRT cells fail against a solid mass.
Okay, let's break those down.
The first is sheer physical burden. A solid tumor is a dense three-dimensional structure. There are simply too many target cells packed too tightly for the T cells to just, chew through efficiently.
That actually brings up a really helpful way to visualize this. A solid tumor is basically a fortified medieval castle, right?
I like that analogy, yeah.
The solid T cells are our invading army, but they don't just march up to the castle walls and start swinging. They get stopped by the second hurdle you were getting at, which is the moat surrounding the castle.
Yes, exactly. In biology, we call this moat the tumor microenvironment, or the TME. And the TME is arguably the most hostile environment in the human body.
Because the tumor isn't just sitting there passively waiting to be attacked.
Right. It actively secretes chemical signals that basically exhaust the incoming T cells. Worse than that, it recruits specific types of immune cells from the host's own body and corrupts them.
It turns our own cells against us.
Exactly. It turns them into guards that patrol the moat and suppress any incoming attacks. So the scalar T cells get bogged down in this chemical and cellular moat, they run out of energy, and the invasion just fails.
Which perfectly sets up the central challenge of this paper. How do we clear the moat so our army can breach the castle?
And Rodriguez and the team, they decided to use radiopharmaceutical therapy, or RPT.
Now, when most of us hear radiation therapy, we immediately picture external beam radiation. A massive machine aiming a high-energy beam from outside the body.
Right, which is like carpet bombing the area.
Yeah. It hits the castle, but it completely damages a lot of the healthy surrounding countryside, too.
And that traditional radiation causes massive off-target toxicity, which, is especially dangerous in pediatric patients. But the RPT used in this study operates on a completely different physical mechanism.
Okay, what did they use?
They used a molecule called copper-67-LLP2A.
Let's translate that out of chemistry speak for you guys listening. The LLP2A part, that's the delivery vehicle, right?
Correct. It is a peptidomimetic ligand. To break that down, peptido refers to proteins, and mimetic means it mimics something.
So it's a synthetic, laboratory-designed molecule built to act like a perfect chemical key for a very specific cellular lock.
Exactly. And that lock is a receptor called VLA4, which just so happens to be heavily expressed on the surface of neural crest-derived tumors, like neuroblastoma.
So we have a synthetic key that will actively hunt down and bind exclusively to the tumor cells. But a key alone doesn't damage the castle. We have to strap a warhead to it.
And the warhead is the first part of that long name, copper-67. It's a radioactive isotope that decays by emitting beta particles.
Beta particles. That's a crucial distinction from traditional radiation, right?
It's very crucial. Beta particles are essentially high-energy electrons. They carry enough destructive force to physically shred cellular DNA, but they travel extremely short distances, often just a few millimeters in human tissue.
Meaning the damage is hyper-localized. You inject this molecule into the bloodstream, it completely ignores healthy tissue, finds the VLA4 locks on the tumor, binds to them, and detonates these short-range beta particles over an extended period of time.
Yes. We have a true targeted smart bomb. It delivers precise destruction without the carpet bombing side effects.
But if we step back and look at the whole strategy here, a massive logistical problem kind of jumps out. The ultimate goal is to send in the living FAR T cells to finish the job.
Right.
If we just dropped a radioactive bomb on the battlefield, aren't we going to irradiate our own elite troops the second they arrive?
That collision is exactly what ruins so many combination therapies. You just cannot administer both at the same time. So the strategy here relied on meticulous, physics-driven choreography.
Choreography meaning the timing had to be perfect.
Exactly. The researchers injected the copper-67 smart bomb, and then they did absolutely nothing for 4 to 5 days. Only after that specific waiting period did they infuse the CAR T cells.
And that window, it must be tied to the decay rate of the isotope, right?
It is entirely dictated by the isotope's half-life. Copper-67 has a half-life of 61.8 hours.
So by waiting roughly 4 to 5 days, you allow the radiation to do its localized damage to the tumor and that surrounding moat, and then naturally decay to a fraction of its original strength.
Precisely. By the time the freshly engineered CAR T cells arrive, the radiation levels are low enough that they don't harm the incoming troops.
Okay, so the timing makes perfect sense. But let's look at the actual dose of the radiation, because my instinct, and I think the instinct of anyone who really wants to obliterate a cancer, is to use the absolute maximum dose of radiation the patient can survive, right? To weaken that castle as much as possible before the army arrives.
It's a totally natural assumption. But the researchers tested that exact assumption in the mice, and the data pushed back hard against it. They discovered this profound Goldilocks effect.
Like finding the dose that's just right.
Exactly. When they used a low-dose 0.5 millicuries of radiation followed by the Cabo T cells, the results were staggering. They saw complete tumor clearance in 75% of the mice in one model.
Wow.
And in another, much more realistic testing scenario called an orthotopic model, the cure rate was 100%.
I really want to pause on the term orthotopic model for you listening, because it is vital for proving this actually works in the real world. Researchers often test cancer drugs by just growing a human tumor under a mouse's skin, right?
Yeah, it's easy to measure, but the neighborhood is just skin. It's not realistic.
Right. An orthotopic model means they grew the neuroblastoma inside the mouse's adrenal gland, which is exactly where this cancer naturally originates in humans.
And growing the tumor in its natural anatomical home means the tumor has the chance to build a highly realistic, fully functional immunosuppressive moat. Clearing a tumor in an orthotopic model is a much, much higher bar.
And at the 0.5 millicurie dose, they cleared 100% of them. That's incredible. But what happened when they cranked up the dose?
They doubled the radiation to 1.0 millicurie, assuming the clearance rate would be even faster. Instead, the cure rate dropped to around 60%.
Wait, really? It dropped?
Yes. And worse, the mice began exhibiting clinical toxicity, specifically significant weight loss and anemia.
That is deeply counterintuitive. Hitting the tumor twice as hard actually protected the tumor in the long run.
Well, it's because exceeding that specific threshold of radiation begins to damage the host's overall systemic health. The higher radiation likely began affecting the bone marrow, and crucially, it compromised the functionality of the very CRT cells they infused days later.
So it proves that this strategy isn't about using brute force to just nuke the tumor. It is about using the absolute minimum effective dose to fundamentally alter the biological rules of engagement.
Perfectly said.
So we have our protocol established a low, precisely timed dose of targeted radiation, a waiting period for the fallout to clear, and then the T-cell invasion. But the most fascinating part of this entire paper is that the researchers found this identical protocol triggered two completely different biological reactions.
Right, depending on the type of tumor they attacked. They applied this protocol to 2 distinct strains of neuroblastoma, and it revealed some incredibly elegant biology.
Okay, let's look at the first strain.
The first strain they tested is called the IMR5 model, and the defining characteristic of IMR5 is that it is highly radiosensitive.
Meaning the tumor cells themselves are physically vulnerable to the beta particles from our smart bomb.
Yes. When you expose IMR5 cells to that low dose of copper-67 in a laboratory setting, the cells slowly begin to die. But the researchers wanted to know what was happening on a molecular level before the cells died. So 72 hours after radiation, they performed RNA sequencing on the tumor cells.
And RNA sequencing is essentially taking a molecular census. It tells us exactly which genetic blueprints the tumor is actively reading, which genes it is turning on, and which it's shutting off.
Exactly. And the census showed massive transcriptional reprogramming. The radiation induced a profound behavioral change in the dying tumor cells. Specifically, they drastically upregulated a genetic pathway responsible for producing a protein called TNF-α.
TNF-α, that's an inflammatory cytokine, right?
It is.
So if we go back to our medieval castle analogy, the beta particles hit the walls and the walls start to crumble. But instead of just collapsing quietly, the physical damage trips a highly sensitive burglar alarm. The tumor itself starts producing this TNF-α alarm signal.
Yes. And importantly, the RNA sequencing proved that the tumor was acting in a paracrine manner.
Paracrine meaning it isn't just an internal alarm. The tumor is literally broadcasting these distress signals outward into the surrounding neighborhood, right into the TME.
Exactly. The tumor is screaming into the void. And when the CAR T cells finally arrive and march into that neighborhood, they encounter this dense cloud of TNF-α.
And what does that do to them?
The researchers proved that this specific signal hyperactivates the T cells. They actually took the liquid that irradiated tumors had been growing in, introduced fresh T cells to it, and watched the T cells whip into an absolute lethal frenzy.
That is wild.
To confirm the mechanism, they genetically engineered a version of the tumor that physically could not produce TNF-α. When they radiated that one, the T cell hyperactivation completely disappeared.
So the brilliance of this is that the low-dose radiation forces the tumor's own stress response to become a beacon for its assassins. The smart bomb turns the tumor against itself.
It really does.
That perfectly explains why the therapy works for a tumor that is weak to radiation. But the second model they tested completely upends that logic, doesn't it?
Oh, entirely. The second strain is the SKNAS model. This neuroblastoma is highly radioresistant.
Okay.
When researchers put these cells in a petri dish and exposed them to the low-dose copper-67, absolutely nothing happened. The cells completely ignored the radiation. Their viability didn't drop a single percentage point.
Okay, if we follow the logic here, this raises a massive contradiction. If the radiation does not physically hurt these tumor cells, then they aren't dying. If they aren't dying, they aren't tripping the TNF-α burglar alarm we just talked about.
Right.
So if you inject the radiation into the mice with this specific tumor, it should fail, because the smart bomb is effectively doing nothing.
That is the logical assumption. Yet when they ran the orthotopic model in live mice, the low-dose radiation followed by CAR-RT cells still resulted in a 100% cure rate.
Wait, in a plastic dish, the radiation did nothing. But inside a living, breathing mouse, that exact same radiation somehow facilitated a total cure.
Yes.
That implies the radiation wasn't actually fighting the cancer cells at all.
That is the exact deduction the researchers made. They realized they were looking at the wrong target. The secret wasn't inside the tumor cells, it was in the surrounding moat.
The TME.
Right. And to prove this, they utilized a much more advanced technique called single-cell RNA sequencing.
Let's clarify the difference for the listener because it's super cool. Standard RNA sequencing is sort of like throwing a piece of the tumor into a blender and tasting the smoothie. You know generally what flavors are in there, but it all blends together.
I love that, yeah.
Single-cell RNA sequencing is like looking at an unblended fruit bowl. You can pick up every individual grape, apple, and banana and examine them separately.
It's a huge technological leap.
Yeah.
It allows you to audit every single individual cell in the environment. They didn't just look at the cancer cells, they looked at every immune cell, every structural cell, the entire neighborhood.
And what did the fruit bowl show them?
The data showed that while the radioresistant tumor cells ignored the radiation, the surrounding tumor microenvironment was radically transformed. The most striking discovery was the severe depletion of cells known as M2-like TAMs.
Tumor-associated macrophages. We mentioned earlier that the tumor recruits corrupt guards. These are the guards, right?
These are the guards. Normally, a macrophage is the immune system's garbage collector. It wanders around eating dead cells and debris. But solid tumors secrete chemicals that essentially brainwash these macrophages, forcing them into an M2-like state.
So once they become M2s, they drop their garbage bags, pick up weapons, and actively suppress any other immune cells that try to enter the area.
They are one of the primary reasons CAR T cells get exhausted and fail. The single-cell data prove that the low-dose beta particles were highly toxic to these specific corrupted guards.
Wow, so the smart bomb didn't dent the castle walls at all, but it completely vaporized the guards patrolling the moat.
Exactly. That's addition by subtraction. We removed the suppression. But the researchers found there was actual addition happening as well.
What do you mean?
To visualize this, they used multiplex fluorescence imaging. This technology allows researchers to assign different glowing colors to different types of cells and see exactly where they are physically positioned within the tissue structure.
So we aren't just seeing what genes are active, we are looking at a literal satellite map of the battlefield.
And the map revealed something extraordinary. In the radioresistant tumors treated with ARC-ET, the environment was completely remodeled into what the researchers called immunological niches.
Immunological niches?
Yeah. Rather than the CAR-RT cells wandering aimlessly through the moat and getting picked off one by one, the imaging showed cytotoxic CD8-positive T cells, the primary killers, and helper CD4-positive T cells physically huddling together.
Huddling together.
Yeah, they were forming highly organized, tight spatial clusters alongside structural cells called myofibroblasts.
So they weren't just invading, they were setting up fortified forward operating bases right inside the moat.
Exactly. The clustering allowed the T cells to communicate and support each other. The RNA data confirmed that these clustered T cells weren't showing the typical signs of terminal exhaustion that usually doom solid tumor therapies.
What were they doing instead?
They had highly upregulated their translational machinery.
Meaning they were actively building the cellular factories required to pump out survival proteins and just keep fighting.
Exactly. The radiation effectively terraformed the moat. It completely cleared the hostile environment and rebuilt it into a supportive landscape where the immune cells could group up, communicate, and systematically dismantle the tumor from within.
This dual mechanism is, hands down, the most elegant part of the study. I mean, think about it. If the tumor is weak to radiation, the targeted smart bomb forces the cancer cells to scream for their own assassins.
Right.
But if the tumor is strong against radiation, the smart bomb simply ignores the cancer, burns away the corrupted guards, and clears a space for the T cells to build their attack formations. It covers both biological bases perfectly.
It highlights how a single, carefully calibrated physical intervention can have drastically different, yet equally therapeutic downstream biological effects, depending entirely on the context of the tumor.
It's a phenomenal result in mouse models, but obviously the ultimate goal is human application specifically, treating children suffering from relapsed neuroblastoma.
And bringing this to the clinic poses a massive real-world problem.
Yeah, you can't just inject radioactive isotopes into a child and hope their specific tumor expresses enough of those VLA4 receptors for the smart bomb to bind to. And doing repeated, highly invasive surgical biopsies on a sick child just to check for a receptor is, well, it's just not an option.
It's not. But that real-world limitation is solved by a rapidly growing field called theranostics.
Therapeutics plus diagnostics.
Exactly. It's the seamless combination of the two. Remember that the LLP2A molecule is simply a delivery vehicle. The brilliant part of this design is that you can swap out the payload depending on what you need it to do.
So before you ever administer the destructive copper-67 warhead, you can attach a different isotope to the exact same delivery vehicle.
Precisely. Clinicians can attach copper-64, which is a diagnostic isotope. It doesn't emit destructive beta particles. It emits positrons, which are entirely harmless but show up brightly on a standard PT scan.
Oh, that's clever. So you inject this diagnostic version into the patient, wait a few hours, and run the scan.
Right. And if the tumor lights up on the screen, it proves definitively that the VLA4 locks are present and the delivery vehicle can successfully reach them.
You can visually confirm the target before you ever fire a shot.
You see what you treat, and you treat what you see. If the scan is dark, you know this therapy won't work for this specific child, and you save them from undergoing a pointless, potentially toxic treatment.
And if the scan lights up?
You simply swap the payload from the diagnostic copper-64 to the therapeutic copper-67, and you administer the drug with total confidence.
It also allows for something called personalized dosimetry, doesn't it?
Yes, it does.
Because you can see exactly how much of the diagnostic drug accumulated in the tumor versus the healthy organs on the scan, doctors can calculate the precise mathematic dose of copper-67 needed for that specific patient.
Exactly. They can ensure they hit that perfect Goldilocks zone we talked about earlier, delivering just enough beta particles to trigger the burglar alarm or clear out the corrupted guards without crossing the threshold into bone marrow toxicity.
It brings a level of personalization, safety, and precision that is desperately needed in pediatric oncology, ensuring the patient's immune system remains robust enough for the Surrocar T cells to do their job days later.
It really is a game changer for combination therapies.
So to bring this all together for you listening, we started with the seemingly impenetrable fortress of a solid tumor protected by a chemical and cellular moat that historically ruins our best engineered immune cells.
Yep, the classic siege.
But by utilizing a highly targeted, low-dose radioactive smart bomb and delivering it exactly 4 to 5 days before the immune cells arrive, researchers found a way to rewrite the rules of that siege.
They either damage the castle walls until the tumor broadcasts a distress signal, or they sweep the moat, eliminate the corrupted guards, and allow the T cells to build forward operating bases.
It truly serves as a masterclass in combination therapy. The CAR T cells alone couldn't clear the solid mass. The low-dose radiation alone couldn't clear it either. But, sequenced with perfect timing, they unlock entirely new, curative biological mechanisms.
And it leaves us with a genuinely provocative thought to explore moving forward.
Oh, absolutely. Because we have seen how a mild, perfectly targeted dose of radiation can so completely disrupt a tumor's local environment that it is forced to roll out the red carpet for our immune system, what other highly localized physical interventions might we be able to use to achieve this same disruption?
Well, if the underlying principle is simply disrupting the tumor's carefully maintained homeostasis to create an opening, radiation is really just one tool in the toolkit.
Right. What if we used targeted high-intensity sound waves to physically vibrate the TME, or highly focused magnetic fields? What about localized temperature changes? Using mild hyperthermia to stress the tumor cells just enough to trigger that same TNF-α alarm. Could we use these non-radioactive physical forces to terraform the battlefield and trick solid tumors into welcoming their own destruction?
It's a huge question. The fields of mechanobiology and physical oncology are just beginning to scratch the surface of those concepts. If the ultimate goal is simply to pull the biological fire alarm, we are definitely going to find many more ways to break the glass.
That is a fascinating frontier to keep an eye on. That wraps up our time for today's deep dive into targeted smart bombs, corrupted immune guards, and the future of CAR T-cell therapy. Thank you so much for joining the conversation. We hope this gave you a deeper understanding of the mechanisms driving the next generation of cancer treatment. Keep questioning the world around you, and we will catch you on the next deep dive.